All right, so we have some notes that we're going to be doing today. So today our notes are going to be on nucleic acid structure and replication. So we're going to be looking at different nucleic acids, including DNA, and then also RNA and ATP. So we'll look at the differences in those structures and also the steps required in replication, how that all works. We're talking about base pairing rules, and then also, the production of proteins. Next week, we'll kind of build off this that we learned this week. So the target, you're going to be able to list the components and give examples of nucleic acids. Make sure you write that target at the top left-hand corner of your notes with your title. Okay, obviously the picture there, that is DNA. That could be used as a drawing for that chart if you would like to. Okay, so nucleotides, we talked about this yesterday. Nucleotides are what make up nucleic acids. There are three pieces to the nucleotide. This picture right here would be perfect for target 1. So target 1 is asking what are the pieces of a nucleotide. Again, we have those nitrogenous bases. If you can see that at the top right-hand corner, we have a sugar, which is a 5-carbon sugar. In this case, it would be ribose. Okay, if it's DNA, it would be deoxyribose. Okay, and then over to the left there, we have a phosphate group. So again, this is something that I would draw on my target right now. This is your picture that you could use for target 1 if you're going for 4 or 5. Perfect picture, perfect description right there. Okay. The nitrogen bases. If we are talking about DNA, okay, we have adenine, guanine, cytosine, and thymine. Again, those are for DNA. The nitrogen bases for DNA. If we are talking about RNA, the thymine gets replaced by uracil. So it would be adenine, guanine, cytosine, and uracil. Okay, that uracil replaces the thymine. Okay, another way to describe these bases, these nitrogenous bases, you can see those structures over to the right. So guanine and adenine would be considered purines, okay, or a double ring structure. And then the pyrimidines or single ring structures would be cytosine and thymine. Again, thymine is a pyrimidine. When it gets replaced by uracil, uracil is also a pyrimidine. Okay, so there they are right there. Okay. The structure, if we look at that 5-carbon sugar, deoxyribose, okay, you can see that that hydrogen is missing an oxygen. That's why we call it deoxyribose. Okay. How many carbons does glucose have? Okay, glucose is a 6-carbon sugar. When we look at deoxyribose, we got that 5-carbon sugar in that ring structure. So that carbon is changed in a different spot in that left-hand corner. Okay. When we look at the structure of DNA itself, okay, when we put all those nucleotides together, we have what's called a double helix, shaped like a twisted ladder. Okay, the phosphate and the sugar make up the rails of that ladder. So you can see what the S and the P are. I don't know if the arrow is going to work here, but the S and the P. Okay, and then in the middle, we have those nitrogenous bases. You can see that we have the T for thymine, A for adenine, G for guanine, C for cytosine. Okay, one other things you might notice, little blue dots, those represent hydrogen bonds. So hydrogen bonds are what hold those nitrogenous bases together. Again, T is going to bond with A, C to G. Okay, that is a target. That's also in your target. So that's the bonding rules for DNA, C to G, T to A. We'll talk about that again here in a second. But when we look at the structure of DNA, it's called a twisted ladder. So it's a two-sided molecule. It's going to have the complementary bases running on each side. And then the backbone of those molecules, or the rails, we made of that alternating sugar phosphate. Okay. DNA, the function of DNA. Okay, this is the big picture kind of going forward. We need to understand this. DNA are the instructions or the blueprint for building proteins. Okay, this is the code. This is what is used. Okay, like the blueprint for your house, it's the instructions, it's the layout for your house. And DNA is the layout, the instructions for making proteins. Okay, specifically when we talk about proteins, we're looking at segments of DNA called genes. But those genes code for specific proteins to give us the instructions for making them. DNA, like I said, is the code. Okay, so when we look at the actual letters in the code, that's when we're talking about those nitrogenous bases. Okay, those letters, the sequence of those letters in a segment of DNA would be called a gene. Okay, again, those are producing those, or those are being used as the instructions to make a specific protein. So genes are specific parts of DNA that code to make a protein. Again, those nitrogenous bases and the order of them are what determine the order of the amino acids, which eventually will be put together to make a protein. Well, a polypeptide and then a protein. So DNA, when we talk about what else it's used to do, so it's copied prior to cell division. Because all of our cells have DNA in them, we need to make sure if we're making new cells that those same cells are made right and that we also have the same DNA in those cells being made. Okay, so when we go through mitosis, which is the process to get to cell division, okay, DNA needs to be copied and then transported from one nucleus to the nucleus of that new cell that's being made. Okay, so you can see here we have the different phases of the cell cycle. I know we haven't talked a lot about the cell cycle yet, but prior to a new cell being made, right after G1, we have S phase, which basically is copying all that DNA. Okay, so we're making these chromosomes are being made, so that DNA is being tightly compacted into chromosomal form, and then prior to a new cell being made, prior to mitosis or M phase, we will have that DNA being copied. Okay. DNA is found in the nucleus of eukaryotic cells. For prokaryotic cells, since they don't have a nucleus, it's not found in the nucleus. It's found as a loop. But they still have DNA. Okay, so when we look at eukaryotic cells, that big blue structure would be the nucleus. You can see it in this form. It's not tightly compacted into chromosome form. All those squiggly lines in that blue nucleus would be that DNA. Okay, and you can see all the other parts of the cell. We have the rough ER, smooth ER. We have some centrioles. This would be an animal cell because it doesn't have a cell wall or have a cell membrane. We have mitochondria there. You can see the little dots represent ribosomes. Those little empty circles probably going to be representative of lysosomes. Okay. Properties of DNA. So some of the things that we talked about. It's double-sided or double-stranded. It's shaped like a ladder. It's got deoxyribose. It's got those 4 nitrogenous bases. It's the blueprints for making proteins. Okay, so all those things. RNA, another nucleic acid. Again, like I said earlier, so one thing about RNA, it's single-stranded, so it's not going to have that other complementary side. The nitrogenous base that replaces thymine is uracil. We're still going to see cytosine, we're still going to see guanine, we're still going to see adenine, but again, instead of the T, it's going to be replaced with a U for uracil. Okay, big difference. Also, the fact that it's single-stranded, another big difference. I think of RNA as kind of the cheap copy. Okay, DNA, is kind of the big blueprint, the big huge molecule. RNA usually is going to be a copy, like that cheap copy of that DNA. Okay, so some things about RNA, again, we'll talk a little bit further here in a second, but know that for the nitrogenous bases, it's still going to have those 3 parts. So we're going to have a phosphate, so we're going to have a sugar, and then it's also going to have those nitrogenous bases with thymine being replaced by uracil. RNA, again, another 5-carbon sugar. You can see in that bottom corner off that 2' carbon. Instead of having just that hydrogen, we're adding the oxygen, so a good way to tell. But it's a 5-carbon sugar ribose instead of deoxyribose. So in RNA, the R stands for ribonucleic acid, so ribose. Okay, and there's your difference there in the sugar. RNA is also found inside the nucleus and outside the nucleus. So it's made in the nucleus, and then it will leave the nucleus to perform the specific function that it has. Okay, depending on what type of RNA it's going to be. If it's mRNA, it's going to be a message. If it's tRNA, it's going to be attaching to amino acids and bringing them to the ribosome. And there are rRNAs, which are basically making up different parts of that ribosome. Okay, but it's found inside and outside the nucleus. Again, here's the types of RNA. So mRNA, M stands for message or messenger RNA, takes DNA outside the nucleus. to the ribosome, takes the message that DNA has, so it's a copy. tRNA, you can see the little squiggly looking dude over there. Okay, that's going to be responsible for getting different amino acids, matching up with the mRNA in the ribosome to put those proteins in the right order, and then rRNA makes up different subunits of the ribosome itself. Okay, so make sure you write those down. These are different functions of RNA. So in your chart, these would be some functions of RNA, and you can be very specific. Again, different types of RNA have different functions. So here's 3 big functions for RNA. So make sure you get those. 3 ways in which DNA differs from RNA. So RNA is going to be single-stranded. RNA is going to have a different nitrogenous base where U replaces T. And then RNA also is going to be found in different locations throughout the cell, depending on what its function is. And then you could also say things about the functions of RNA versus DNA. Okay, so those are some differences that we see. ATP, so the third nucleic acid, ATP, so adenosine triphosphate. Again, we know that the function of ATP is that usable chemical energy. Okay, that energy is held in the bonds of the phosphate groups. If we look at this nucleotide, it's also going to be quite a bit different than DNA and RNA. Obviously, the sugar is the same as RNA, so the sugar is ribose. It's only going to have one possible nitrogenous base, and that's going to be adenine. Okay, and then the phosphate group, so the triphosphate, and in the other 2 nucleotides that we looked at, there's only 1 phosphate group. With ATP, there's 3 phosphate groups. So again, that's where that energy is held. Okay, in those bonds between the phosphates. So ATP triphosphate, 3 phosphate. Okay. Also, these don't bond together, so we're not going to see a bunch of nucleotides making up a polymer for this one. That's why we call ATP a modified nucleotide. And again, because they're not going to be bonded together to make these bigger molecules. Okay, so there is no real polymer for ATP. ATP is just what it is. It's just that small molecule that's set up like this. Okay, what are the 3 parts of a nucleotide? We've already answered that, but hopefully you can. I would write this question down. Just make sure, again, this is a target. And again, give 3 examples of nucleic acids, target 2. Okay, this should not be a problem. So we have the nitrogenous base, sugar, and then we have the phosphate group. We have different nucleic acids, ATP, RNA, DNA. Okay, now moving on to DNA replication. So like we said, DNA needs to be replicated before cell division. Okay, so we need to have a process where that takes place, and there needs to be rules where this would happen. Okay, so this would be another target. If you wanted to write this down, you could. Again, this is just the targets from our weekly targets, but we're going to list the steps of DNA replication. We're going to put them in the right order, and then explain how complementary strand is built through what we call semi-conservative replication. Okay, so we're looking at how are we building a whole new strand of DNA from an existing one. Okay, so step 1. When we look at the process for replication, we have that DNA molecule needs to be unwound. So those hydrogen bonds that are made between the nitrogenous bases need to be broke. Okay, there's an enzyme that is responsible for this. Again, this is a protein. So helicase, what it does, it goes through and it breaks those hydrogen bonds between the nucleotides. Okay, basically you want to think of it as it's the thing that unzips DNA. Okay, it breaks those bonds, think of it like a zipper. It's going through and it's unzipping. Okay. After that happens, okay, there's different other proteins that will go through and hold that part of the DNA molecule that's being unzipped, hold it apart. Okay, so these are those binding proteins, and they basically just act to hold the strand open. So they don't want it to. Those hydrogen bonds are going to want to come back together to reform, and these proteins bind to each strand, opposite strands of the DNA, to keep them from doing that. Okay. Step 3, yet another protein, DNA polymerase 3, very important for replication as well as other processes. Is this DNA polymerase 3? It starts to go through, and where we see that molecule, a DNA molecule unzipped, it's going to lay down what we call complementary base pairs. So where there's an A, it's going to lay down a T. Where there's a C, it's going to lay down a G. Where there's a G, they're going to lay down a C, okay, and so on and so forth. Okay, Again, DNA polymerase 3, again, when we talk about DNA polymerase 3, we're making a whole new strand of DNA based on that existing DNA strand. Okay? And these will be working in the 3' to 5' direction. And you can see that in that diagram there. Okay? Everything, I just want to make sure, yes. Okay, so it begins laying down polymerase based on an RNA primer. So we are going to have an RNA primer, just kind of signals where to start, where that DNA polymerase is going to start laying down bases. When we talk about base pairing rules, so this is target 3, I believe. The base pairing rules will be A to T, C to G, G to C, T to A. Okay, they go back, so they're complementary of one another. So in the diagram there, you can see every G is bonded with a C, every C is bonded with a G, every T is bonded with an A, every A is bonded with a T. Okay, so for DNA, the base pairing rules are just like you see. A to T, C to G, T to A, G to C. Okay, they work back and forth. Okay, so again, this DNA polymerase is going to come through. It's going to lay down the correct bases according to what that other strand is. So what that first strand looks like. That new strand that's being made is going to get the complementary bases that go with it. Okay. Step 4, when we look at the Okay, so back with you real quick. Just wanted to kind of go over things. So DNA ligase is an enzyme that's going to link what we call Okazaki fragments on the lagging strand of the DNA. So this is just an enzyme that kind of goes back on the lagging strand and takes these sections of DNA and basically lays them down in the opposite direction of that lagging strand. Okay, so just the last segment, it's just another part of the process. Okay. Replication bubbles. So when we look at DNA, because it's such a huge molecule, we are seeing this replication process happening in different spots along that molecule. Okay, if we were trying to go from just one end and work all the way across, it would take a lot longer than if we have what's called these replication bubbles, where we can see this DNA replication process happening kind of in those different spots to make it go a lot faster. Okay. Replication practice. So when we look at this segment of DNA, this would be like a very small gene maybe, we're going to see if we can lay down the complementary strands. So I'm setting you up. This would be one side of that DNA molecule, and you are going to be acting as DNA polymerase 3, and you're going to be laying down the complementary strand based on DNA pairing rules. So for every A, there's going to be what? For every A, that polymerase is going to bring another what? C, what's going to happen? So let's see if we can do this. So take a second. See if you can figure out what that complementary strand of DNA would look like. Okay, and there it is. So again, you can see wherever there's a C, that polymerase 3 is going to be laying down a G. Wherever there's a G, bring in a C. Wherever there's a T, there's an A. Wherever there's an A, there's a T. Okay, so again, that complementary strand, that's what it's going to look like. So this is our new strand being made, the one in red. Okay, that original strand is the one in black. Okay, that's that one that DNA polymerase 3 is going to be reading and then laying down those complementary bases. Okay, as of right now, so protein synthesis, we're not going to get into that. We're going to stop here because that's replication. So hopefully that was helpful. If you have any questions, please write them down. I know for a third period, I won't be here tomorrow to go over this. So if you have questions for me, please have them ready for me and I'll come in ready to answer them. All right, thank you.